Dear viewers, greetings. In this video, we are going to see the various types of fluorescence spectroscopy. Fluorescence spectroscopy can be classified into several types based on different techniques and applications. These types enable a range of studies from basic fluorescence properties to complex molecular interactions. Here are the major types of fluorescence spectroscopy: steady-state fluorescence spectroscopy, time-resolved fluorescence spectroscopy or TRFS, fluorescence anisotropy or polarization spectroscopy, fluorescence resonance energy transfer or FRET, fluorescence lifetime imaging microscopy or FLIM, total integral reflection fluorescence or TIRF spectroscopy, multiphoton fluorescence spectroscopy, conventional or emission fluorescence spectroscopy. The first type of fluorescence spectroscopy is steady-state fluorescence spectroscopy. In steady-state fluorescence spectroscopy, the sample is continuously excited with light and emission spectrum is measured at equilibrium. The fluorescence intensity is recorded as a function of the wavelength of the emitted light. Applications: steady-state fluorescence spectroscopy is commonly used to study the overall fluorescence properties of the sample, such as emission spectra, quantum yield, and environmental effect of the fluorophores. The second type of fluorescence spectroscopy is time-resolved fluorescence spectroscopy. In short form, it is called as TRFS. In time-resolved fluorescence spectroscopy, the decay of fluorescent intensity is measured over time after a short, intense pulse of excitation light. The fluorescence lifetime, that is, the time taken for the excited fluorophores to return to their ground state, is analyzed by using this time-resolved fluorescence spectroscopy. Applications: this time-resolved fluorescence spectroscopy or TRFS is used to study the dynamic process in molecules such as energy transfer, molecular interactions, and conformational changes. It provides information that steady-state fluorescence cannot offer, such as decay kinetics and lifetime measurements. The third type of the fluorescence spectroscopy is fluorescence anisotropy or polarization spectroscopy. This technique measures the polarization of emitted light from a fluorophore after it has been excited with polarized light. The degree of fluorescence anisotropy is dependent on the rotational mobility of the fluorophore. Applications: the fluorescence anisotropy is often used to study the size, shape, and binding interaction of molecules, particularly in protein ligand binding studies and molecular mobility investigations. The fourth type of the fluorescence spectroscopy is fluorescence resonance energy transfer. In short form, it is called as FRET. The FRET occurs when energy is transferred non-radiatively from an excited door fluorophore to an acceptor fluorophore in close approximately 1 to 10 nanometer. The efficiency of this energy transfer depends on the distance between the two molecules. Regarding applications, the fluorescence resonance energy transfer or FRET is a powerful tool for studying molecular interactions, protein foldings, conformational changes, and distances at the nanometer scale in the biological systems. The fifth type of fluorescence spectroscopy is fluorescence lifetime imaging microscopy. In short form, it is called as FLIM. The fluorescence lifetime imaging microscopy measures the fluorescence lifetime of molecules in a microscopic field. It generates an image based on the differences in the lifetime of fluorescence across a sample rather than fluorescent intensity. Regarding applications, fluorescence lifetime imaging microscopy is widely used in cell biology and medical research to study the local environment of fluorophores in living cells, such as pH, ion concentration, and oxygen levels. The sixth type of fluorescence spectroscopy is total internal reflection fluorescence spectroscopy. In short form, it is called as TIRF spectroscopy. The TIRF spectroscopy involves illuminating a sample at an angle that causes total internal reflection at the interface between a glass slide and the sample of a solution. This results in an emission wave that excites fluorophores close to the interface, typically within 100 to 200 nanometers. Regarding applications, TIRF spectroscopy is highly sensitive for studying surface-bound molecules and processes at cell membranes such as receptor-ligand interactions, single molecule detection, and molecular dynamics at the membrane. The seventh type of the fluorescence spectroscopy is multiphoton fluorescence spectroscopy. In multiphoton fluorescence spectroscopy, two or more photons of lower energy, typically near infrared, are simultaneously absorbed by a fluorophore and exciting it to a higher energy state. The emission occurs as if the fluorophore absorbed a single photon of higher energy. Regarding applications, this multiphoton fluorescence spectroscopy technique is often used in deep tissue imaging and live cell microscopy due to reduced photo damage and better tissue penetration. The final and eighth type of fluorescence spectroscopy is conventional or emission fluorescence spectroscopy. This conventional fluorescence spectroscopy is the basic form of fluorescence spectroscopy where the sample is excited with a specific wavelength and the emitted fluorescence is measured as a function of wavelength. Regarding applications, this conventional fluorescence spectroscopy is used for general fluorescence studies such as determining emission spectra, investigating molecular environments, and studying fluorescence quenching and enhancement. Dear viewers, that's all about the various types of fluorescence spectroscopy. Thank you for your support. Thank you.